Hi, I'm Charles Wyckoff, and it's an absolute pleasure and privilege to be here with Dr. Samuel Barone, Chief Medical Officer of Gemini Therapeutics. He has a fascinating history, having served as a medical officer at the FDA. He's been CMO for multiple biotechnology companies and served as a flight surgeon in the U.S. Air Force. Sam, thanks for being here today.
Well, thanks for having me, Charlie. It's a privilege.
So for every retina specialist out there, we're acutely aware of the challenges associated with sort of AMD and AMD management, and in particular, the huge unmet need globally for a treatment, a strategy to approach patients with geographic atrophy. And fortunately for patients and this whole space in general, there's a tremendous amount of activity and energy right now going on to looking at possible therapeutics that could bring benefit to patients. And so in that context, Sam, tell us about Gemini. Tell us about your lead product right now, Gem 103, and tell us what differentiates you in this space today.
Yeah, very, very happy to do so. So as a retina specialist myself, it is very meaningful to be working in this space and to be addressing the needs for those patients. And it's very exciting that there's so much progress and the ability of people addressing it, right? Addressing geographic atrophy and the dry macular degeneration space. So what we're doing in Gemini, so we're a complement company as well, which many of the lead programs are addressing the complement cascade, and because it's been very much implicated in the causation of dry macular degeneration and geographic atrophy. But what separates us and what gets me excited is we're really doing it with a precision medicine approach. So we know that AMD is multifactorial, but there is about a 50% genetic component to it. And so that by looking at the individuals who have specific mutations, particularly in the complement factor H gene, then those individuals are at higher risk for developing macular degeneration. That by targeting those specific individuals, that we can have an advantage and we can make a bigger difference.
So let's unpack that a little bit, because I think there's a lot in what you just said, and it's super important to the field. And obviously, there's been this long-term understanding now over the last 15 years and growing about the role of complement and how people hypothesize that maybe 50 or even 70% of my risk of getting this disease is baked into my genes. But CFH from the beginning was one of the first ones I think identified in a couple of science papers. What's the hypothesis of why these haplotypes, specifically in CFH, are associated with AMD? Is it sort of the early stages of AMD? Is it rate of GA progression? What is it that's associating complement factor H haplotypes with AMD specifically?
Right. So complement factor H, I mean, more broadly, complement factor H is one of probably the most important regulator of the complement system in the alternative pathway, right? So when there's a mutation in the gene for complement factor H, you're going to see impact right downstream. So in terms of numbers or in terms of odds ratio, I think it's sort of important to look at the most common complement factor H mutation is homozygous and 402 loci, 402HH is what it's referred to as. And it's about 40% of the population with AMD has a 402HH mutation. And that's been shown to give about a 7 or 8 times odds ratio in terms of developing geographic atrophy. So it's actually the development of the geographic atrophy to get to that space. The heterozygous allele for 402H, so 402HY, that has about half the amount of the risk at about 3.5 to 4. And just to put that in perspective, the odds ratio for like developing heart disease with hypertension is around 3 to 4 as well.
Right.
So so I think that provides a lot of context to say that, particularly with the HH mutations, that there's a very high risk if you have this mutation that you will develop geographic atrophy.
And what's the connection there between the HH sequence and is it that the common factor H in these patients has less function? Is there lower expression levels? Like what's the phenotype associated with that genetic variant?
So with the 402HH, it's what actually ends up happening. It's a protein is still expressed, so it's not but it's not functional. There's some dysfunction into it or in it. And what it actually has to do, with a binding domain of the 402, or sorry, of the complement factor H. So there is still some function around, but its ability to localize to membranes. And particularly like on the RPE cells, that's
Right.
where it's limited with the mutation.
Fascinating. Okay, so then the approach currently with Gem 103 is to produce sort of exogenous CFH and then give it by intravitreal injections. Is it a complex molecule? Is it hard to produce? Like, how are you making it, and how do you purify it?
So the idea of replacing complement factor H has been around for several years because, as you said, was identified 15 years ago at the highest risk, or the high risk with complement factor H. The challenge has been that it requires the full length complement factor H to be able to make a difference. So they've looked at, hey, like, well, what if we just deliver the subunit, for example, that is
Right.
that then compensates for where the mutation is, or looking at different subunits, because the full length recombinant human complement factor H has been a challenge. And that's actually in animal studies, that's been shown not to be as effective as the full length molecule. So what we've been able to do, or the Gemini success, and some of this predates me, is our ability to manufacture the recombinant human complement factor H. No one else has been able to do it. And so that's sort of our special sauce, that we've been able to make the full length protein that will fold just like in an endogenous state, and then be able to have an effect.
Fascinating. And then before we get into the data that you guys recently presented and released, tell us then, so you inject the CFH into a patient's eye, and what's the hypothesis of then what happens to the complement cascade after injecting that?
So in the terminal complement, so where complement factor H factors in, it's right at the level of C3 and C3 convertase, or C3b convertase, right? So that's where the impact is, and it's the natural regulator. As I mentioned before, it's a very important regulator within the system. So in contrast to the other some of the other programs that are delivering an inhibitor that is something that's artificial, this is the natural regulator of the system that we're putting in, and it impacts, right, like I said, right at the level of C3 convertase, to be able to impact anything downstream. Now, in addition to that, so that's just within the terminal complement, there's additional things that complement factor H can do regarding supporting phagocytosis and repairing oxidative damage. So there's some other off-target effects that also can be helpful with complement factor H.
Fascinating. And then to pressure test that sort of role in the actual sort of complement cascade itself, what we think of traditionally as the alternate or the other two activation pathways, C3, C5, MAC. So you inject it. Is it overly simplistic to think that you're down regulating that sort of transition of C3 convertase to C5 convertase and then MAC? Is that overly simplistic? Are we doing something different than just sort of slowing down the transition of the pathway forward?
That's a simple way to look at it, but I wouldn't say overly simplistic, because I think that's just a component of it, right? So we will have that impact and be able to downregulate things into the full membrane attack complex, and we can get into some of the biomarker data that we have that supports the evidence of that. But then in addition, there's the off-target effects that are a bit harder to quantify, and from a regulatory and drug development perspective, I don't think that the that hasn't caught up to being able to prove that we're doing those things yet, but it's something that gets me excited too, to think about like these additional off-target effects and our true ability to protect cells before we get to geographic atrophy.
Right, it's fascinating. I feel like the more I learn about the complement cascade, and I'm not a complement specialist, but the more I realize I just don't understand. It's amazing how many roles each of these little molecules seems to play. It's fascinating cascade. Thank you. So, okay, let's move forward into your data. So recently, I think just a couple of weeks ago, middle or end of June, you guys had a big news release where you released some data related to your ongoing phase 2A REGATTA study. And from my perspective, there seemed to be some clear positive signals there, but then there were some signals that were a little more challenging to digest, at least for us, sort of looking at GA growth rates and sort of what we're used to seeing with a couple other programs. So tell us about the data from your perspective. Maybe we'll start with who was the patient population, what was the study design, and then we'll walk us into what you found.
Yeah, I think that's the key is about the thinking about the trial design, right? So it was set up as a safety study, right? With 62 patients, open label, so no control group, and really, and with some patients. This was, so let me just take a step back too. As we had a phase 1 that had been read out previously, and that had involved 12 patients. So we had only dosed 12 patients before this. So right. This, when this was initiated as a phase 2A safety study, there was, as and being an investigator, until it's proven to be safe, or more, even increasing your comfort level to be safe, there's going to be, you're going to enroll the most severe patients, right? Or the most advanced patients, right? So the first patients that got enrolled in the study, and then the ones that we reported who made it to the 6-month time point, those are the patients who had the most advanced disease. So in general, it's the patient population. When you look at the numbers, it's in general, what you would expect from an AMD population, but it's individuals who there is a skewing, if you will, more toward more advanced disease. So right. Poorer vision, larger lesions, and more uniform, or sorry, more central, right? So more involving the fovea. So we saw that was sort of at first, and why it's just, an important caveat whenever you think about looking at data, looking at even comparing to different trials. What actually am I comparing, and how different are the characteristics or the groups that we're looking at?
And then the genotypes of these patients, did they all have these complement factor H mutations, these changes, or was it a general population?
So there was about 2/3 of them who ended up having the 402HH mutation. So 43 out of the 62 had a 402HH.
Okay, and then how were patients dosed?
The patients were dosed in 2 cohorts. So the first cohort, they were dosed at a 250 microgram dose intravitreally monthly. We followed them for 3 months, and once a 3-month once a 3-month safety signal was observed, or establishment of safety after 3 months, then there was the opportunity to escalate individuals to a 500 microgram dose. At the same time, we started a second cohort, at which time individuals were just started at 500 right from the beginning, right? So 2 cohorts, 250 escalated to 500 after 3 months, and then the second cohort had 500 micrograms starting at baseline, and repeat monthly injections.
And then we'll get into the data in just a moment. But then, so you released 6-month data recently. But were patients able to continue on with dosing? How long of a study is it? What's the final duration of it?
It's a great point to clarify. So it was individuals were initially consented for 6 months, and then they have the opportunity to extend for an additional 12 months afterwards. And thus far, I mean, thus far, it's been a really great rate of compliance. And individuals staying on the treatment, and I think that shows a lot, not just our commitment, but the commitment of the patients and the investigators to keep patients on it.
It's a great subtle point you bring up there, a really important one. And I think we see that across many of the GA programs, how these patients in particular, remarkably compliant. I think it shows that they're significant symptoms. They're interested in things to make them better, even as you say, in a population like this with large unifocal lesions, because all the lesions, most of them have coalesced, and these large, they still want something that slows the progression of this disease. I think it shows the unmet need here. And then just one more question about that. So after 6 months, before 12 months, patients are continuing to receive monthly dosing at the 500 level, is the plan?
Correct.
Okay. Okay, now then walk us through the data that you saw. We get anatomic endpoints, you have the biomarker data. The biomarker data is fascinating. I want to hear details about that, but give us sort of your perspective on what the data meant and what it looks like.
Well, first and foremost, this was a safety study, and that's what we set out to show. And I think we've really checked that box. I'm really happy with the safety that we've seen. There were no SAEs related to the drug. There were no ocular SAEs. There were no ocular AEs that related to someone discontinuing the study or even missing a dose because of an AE. So a really great profile from a safety perspective. The total exposure at this, and this is just a snapshot, so that's an important thing to note, that this is just a data snapshot. So patients continue, and we're going to have additional data to present. But the exposure was over 28 patient years of the monthly dosing. So a great achievement for our company, right? We had only done 12 patients a single dose before, and now we have over 28 over 28 patient years. So that's a great thing for us.
Awesome.
As a retina specialist, and just we sort of hit on this a little bit earlier, or maybe it was in our pre-discussion, talking about some of the inflammation that can occur. But right, this is an important thing that we need to be looking for and vigilant about in the space. So we had no endophthalmitis, we had no vitritis, no retinal vasculitis, no vascular occlusive events. So great stuff to be seeing in the back of the eye. There was mild iritis, which was less than or equal to 1+ cell. So it was 0.5 cell in two patients and 1+ in another. That was observed in 3 patients overall. It didn't lead to any interruption in GEM-103 therapy. They actually, 2 of them didn't even, the investigator didn't even feel the need to treat. It was so mild, and just brought them back at the next month and said, we'll continue to give your treatment, no interruption in therapy. Good. So great from the inflammation standpoint as well. Good.
Yep.
And then the final thing, I think, when we put this into the bucket of safety is the idea of the potential for increased risk of CNV, because that's been seen in some of the other programs that are in the complement space. At baseline, we had 31% of the patients had a history of CNV in the fellow eye. Right. So as you know, that's going to increase their risk of CNV in the study eye. We only saw 1 case of wet AMD. It was the investigator saw a macular hemorrhage in one patient at month 1. So if there was something there, it was brewing, earlier. Yeah. The patient had a history of wet AMD in the fellow eye. The patient then got treated with anti-VEGF, and continues on the trial. The reading center, and everything is reviewed by a reading center as well. The reading center actually didn't even see CNV, so it was not even confirmed by the reading center. But I put it in there. We count it. If the patient got treated, we got a macular hemorrhage. We want to err on the side of caution when looking at for safety signals.
Right, fascinating. Okay, very helpful to hear that safety picture. Sounds very clean. And then tell us about the anatomic data first, the geographic atrophy area over time. Did you expect to see a difference here between the fellow eye? I know there was obviously no sort of sham control arm. It's an open label study, so we're comparing to fellow eye growth rates. Some people think that that's optimal because you have a normalized genetic background. Some people think that can be confusing and it's better to use a sham control. Safety study, as you pointed out, really important point, but did you expect to see a signal in growth rates?
I think at this early, we would have loved to have seen one, right?
Sure.
But I think at this, at this early time point, it's not expected.
Yeah.
I think with,
Yeah.
when you look at the slow progression of the disease and just the way that other people, other studies, other active dosing studies, even other natural history studies, they're looking at the progression of geographic atrophy over much longer periods of time, or much more extended intervals between between following up. So
Yeah.
our time points are 3 months for all the patients, and then half the patients at 6 months. And there's just not much that's out there that, in terms of numbers, what does the progression look like at a 3-month time point, right?
And it's
so
critical what you just said. It only half the patients at 6 months also.
yeah.
Okay.
So there's still much more that we're continuing to collect data, and there's still much more that we're looking forward to seeing. And I said, and then also thinking about the patients who made it to 6 months, only half of them, right? And it is those most advanced patients, right? So at the beginning, right, and then that is going to tend to drift over time. But so there's a lot of challenges from that perspective, and I just think that it's too early to tell or to say anything definitive from the geographic atrophy perspective. Also, in the fellow eye comparison, that's there's you can look at it as pluses and minuses. I mean, I think the big thing is that's not accepted as a regulatory to support in a regulatory approval, right? So that's never the ability to do that. So it's not optimal from that perspective, and looking for signals. And then I think some of the other thing with the fellow eye and the way we presented the data here is that that we've shared the data. It's that it's exactly the same, the patient. So we just tried to be most inclusive and show, hey, the top line data, but those fellow eye aren't necessarily matched to the study eye group. So we continue to look more at the data, look at things with, for example, better matching, or just using the contralateral eye, for example, looking at some of the genotypes, and looking for different evidence there. And we look forward to sharing more of these data at the fall meetings.
Awesome. And the last question about this data set is the biomarker data. I really sort of congratulate you and your team for being robust in your biomarker analysis. I think this space could really benefit from sort of detailed understanding of what are we affecting with all of these different therapeutics on this complicated cascade. And we've seen very little of that data out of multiple programs now looking at this. And you guys presented quite a bit of biomarker data. Tell us your thought process there behind that. What does that mean to us?
Yeah, so I've only been with Gemini for just a little bit over 2 months, and when through the process of in talking to them, this is one of the things that got me most excited, was looking at the science the way that we've committed to do so. And again, this also speaks to the earlier point that we were talking about, the patients staying on study. All of our patients, they got aqueous humor taps at every visit. So what they got their monthly injection, all the patients also got aqueous humor taps. So a great amount of data for us to be able to look at, and really for us to be able to say, we're committed to the science and understanding exactly what's going on in these patients. Some of them are exploratory because there's not a lot. There's not a lot that's been out there about in terms of what are how in an active treatment group, what is the implication and what's actually happening with some of these biomarkers. So some of it is exploratory, and we weren't exactly sure what we were going to see, but we're very excited about the results that we have received. So we looked at the two most important ones are BA and C3A, which are the direct breakdown products from the C3 convertase, right? So what we saw with the GEM103, our complement factor H replacement, is that we saw a dose response that it was sustained over time, and the markers reduced to approximately 40% in the BA and the 20% in the C3A. So just to me, this is saying, like, at the physiologic level, like, we are doing something, and it shows that we are.
Fascinating. I think that's quite meaningful to the field. And so what happens next with GEM103? You have this ongoing phase 2A study. You're going to have more data coming out, which we all look forward to, but what's the next step?
Yeah, so in addition to the data that we've received now, I mean, this gives us a lot of confidence moving forward, that we have a safe product, and that we're doing something at the physiologic level, and that the GA will follow. So we're right now in the process of ramping up for that randomized controlled study that's going to be able to show the true benefit of complement factor H. We're in the process of discussing with regulators to continue to refine the trial design. We anticipate that it's going to be able to be powered such that it could serve as a pivotal when it's successful. And we also look forward to looking to be able to leverage some of the things that we feel are truly differentiating about complement factor H. So we're really looking at and leveraging the precision medicine approach. And then we also feel that based on these biomarker data, the biomarker data that we've seen thus far, that we are confident that we can have a extended dosing frequency in between injections, and so we will also be exploring something additional than monthly.
Exciting, a lot going on. So maybe I'll broaden the discussion here just a bit. You guys are at a fantastic, very exciting stage of development, but there's of course a couple other programs further down the road, if you will. And the one closest to sort of a very important finish line, especially from a patient perspective, is of course the Derby and Oaks phase 3 trial program that we anticipate data publicly sort of later this quarter, actually, in 2021. So what are your expectations of what that data is going to look like, right? We had this failed program in lampalizumab so many years ago that's still relatively painful for the entire field to think about, right? So much energy went into that, and there was so much pent up need. There's more need than ever, but there was a lot then, a lot of interest, and it failed. And so what do you think we're going to see with Derby and Oaks, and how are those results going to impact your development?
So, I'll defer to them about what, sort of the expectation to see. But I think, based on what based on the early data, the way that Philly trialed, so their phase 2 Philly trial, I think that there's a lot of potential there for success in Derby and Oaks. I think it's great for the field. I think it's just going to continue to generate more interest, generate enthusiasm, generate investment for further further exploration. I think it's a great step. In terms of us, it would their success would benefit us too, right? Because just for those very reasons, like more investment, more enthusiasm in the space, more doctors are going to be interested in being involved in additional trials or looking for what's next. patients, as well. There's going to be a greater awareness that something is out there and potentially out there for them. So it's something that's very helpful to us. And then I think, like I said, we look for ways that there's potential opportunities for improvement, because it's reducing the rate. If it carries through to compared to what we saw in Philly, it's reducing the rate of progression, but things are still progressing, right? So there's a lot of room for improvement. Does it going to have to be monthly injections versus is there an opportunity for some kind of increased dose frequency to be able to be administered? What's the role, like I said, in a precision medicine? What's the role of in a population who doesn't have a functional complement factor H? If we replace that functional complement factor H, are we going to be able to see a better treatment effect than the 30% rate of reduction? So there's a lot of unknowns. But I think it's great for the field overall.
All fantastic points. Maybe one last parting question for you, and we'll broaden even more here.
Okay.
So you've got a fascinating history, right? You've been heavily involved in the science of many different companies over the years, right? And you work directly with and for the FDA. You have sort of fantastic perspectives. And the one thing I'd like to ask about is gene therapy, right? So we have a lot of activity right now in the space looking at gene therapy. A lot of it in inherited retinal diseases, the monogenetic things, retinitis pigmentosa, and the many different subtypes there. You've got the X-stated retinal diseases producing an anti-VEGF molecule inside the eye. You've got some other complement companies looking at gene therapy, a lot of interest in gene therapy. And you were specifically the CMO previously for companies focused on gene therapy. So what's your perspective on the ongoing work in retina right now related to gene therapy?
Yeah, I think it's excellent as well, and I'm excited to see the progress that continues to be made along those fronts. I mean, I started working with retina gene therapy in 2009. So there's improvements there. Late phase studies. There's an approval with Luxturna from Spark, like in late phase studies. And so, we're continuing to make progress, and all that is just, it's outstanding to see. But additionally, I think what it also shows, and what we've seen through some of these different programs, that there's still a lot to learn, right? Mm-hmm. So there's different challenges that continue to present regarding inflammatory reactions. The sort of I think GenSight they're it's a bit of a head scratcher about what's going on in the fellow eye in their program. So there's still a lot to learn, and I think the most important thing that people looking at it until it's sort of ready for prime time, or just sort of thinking about it as these things continue to progress, is just remembering that it's not just gene therapy and everything is the same under gene therapy. And that we have to look at in each indication, there's going to be separate nuances and separate things to consider. Adverum is a great example. They really show the ability to, with their intravitreal delivery, to be able to decrease the need for frequent anti-VEGF injections in wet AMD. But then in the diabetic space, they had this big inflammatory response, unfortunately. So, is that just in diabetic eye disease, where you have maybe a larger impact or larger decrease on the blood retinal barrier that allows some of these inflammatory factors into the eye, and it may be safer. Intravitreal gene therapy still may be safe in certain indications. I mean, these are all part of the nuances, and people just need to remember when they look, and not just like think of it all as one. It's like, oh, that's just gene therapy, and there's not. I think it's extremely
No, very well said. Couldn't agree more. So much to learn, so much excitement. Well, Sam, I enjoyed the conversation. Really put you on the spot there in a lot of ways. Appreciate your insights. Appreciate the work that you're doing. You're moving our space forward. Thank you for what you're doing, and look forward to continuing the conversation.
Thank you, Charlie.
Have a great day.
You too.